Genetic drift is 100% completely random changing, statistically random change in the allele frequency of a population based on selection. So think about this for a minute. There are a couple of examples of types of genetic drift that we'll talk about. But before we go there to look at two specific examples, imagining or trying to visualize how it is random and statistical. And to me, the best examples come from imagining a population is a bunch of marbles. Because you could do this, or candies. You could have a pile of candies and you randomly reach in, and let's just say that our starting, whatever quantity, proportion of our population is half and half brown, half green. Genetic drift is when the reproduction, the change in allele frequency, if you were to draw 10 marbles out of here, you aren't necessarily going to draw half green, half brown, even though what's in the bag is half green, half brown, right? You can reach in and there's a chance you could draw all brown marbles. That's random. That's statistical random madness. But then you'd have all brown marbles and the population would have changed because of random picking. What? Random picking. That's it. If you imagine, like, look at this. If you drew 6 brown and 4 green and you made a new population that now has more browns than greens, and then you drew again and made a new population that has 7 browns for every 3 greens, which is 70 brown in this bag and 30 green. It's random. You would expect to draw 7 brown and 3 green if you pull 10 marbles out. Genetic drift says, good luck. And if you draw something different, like 4 brown and 6 green, your luck is different, right? Go buy a lottery ticket because that's how lottery tickets work. It's random. It's random. And now you have a population that has changed because of this random draw. 2 flavors of genetic drift, random draw. One of them is called a bottleneck. And guess what? Our little cutie patootie cheetahs experienced a bottleneck back in the day. A bottleneck occurs when some sort of usually traumatic event, a volcano goes off and 90% of all the critters are game overed. The 10% that are remaining are like a little draw. So here you have second generation and the little draw, like we just did a tiny draw. We didn't draw 10, we actually only drew 3. And then we repopulated from only 3 critters. If a volcano killed all the cheetahs, whatever 10% of cheetahs were left, number one, that's not a lot of genetic diversity. True for cheetahs. Something happened to those guys because they're really, really similar, genetically similar to each other. And they think it was a bottleneck event that caused this reduced genetic diversity. The resulting population can be totally different. And again, right? Random. How did this happen? It was random. That's a bottleneck. Did I say bottleneck or did I say bottlenecked? I said bottleneck. Of course I did. Wait a second. There's one more. It's called the founder's effect. The founder's effect is when this little group, it's not an actual event that takes place. It's that a little group, a little random group goes and they leave and they're like, bye-bye, cheetah friends. We're going to found a new place. And that new place that they are going to found, they have a allele frequency that's different than the original population. It's a change in allele frequency that happens because a small group split off and went and did their own thing. Founder's effect is different than migration. If that group were moving to an existing population, that's just gene flow, that's migration. But if they're moving and they're establishing their own, they're not adding their alleles into an existing pool, they are the pool. That is called the founder's effect. Okay, and founder's effect and bottleneck are examples of genetic drift. Now, we have 4 strategies, 4 mechanisms of evolution. Microevolution, we have 4 mechanisms. What about speciation? What has to happen in order for speciation to take place?